Part 2 of the case study is going to involve focusing in on the therapeutic potential of treating the epigenome. So what we're going to look at is leukemia. Now, it will appear on first reading or first run through to be complicated, but take your time, read it carefully, and then make your own notes as to what you think is going on. So, histone H3K4 methylation, including mono, di and trimethylation, has been found generally to be a biomarker for active gene transcription. So that means a gene is turned on in the presence of the methylation signals. There is a notable prominence of epigenetic dysregulation in acute myeloid leukemia, driven by the MLL gene. Dysregulated histone lysine methylations can lead to a disease such as cancer. For example, 75% of acute leukemia in infants and 10% of that in adults contain an MLL gene translocation. A translocation is where the gene is shifted along the reading frame. It's moved in within the chromosome. This type of subtype of leukemia shows a very poor prognosis. MLL stands for mixed lineage leukemia, MLL, a large multi-domain protein. It's also the MLL gene has a gene product, MLL, and it's a large multi-domain protein containing 4,000 amino acids. And this multi-domain protein can methylate H3K4. And it has a C-terminal set domain. It doesn't matter what SET stands for. It's a set domain, and this set domain is catalytically active as a histone methyl transferase. So that means it puts methyl signals on. In addition, cellular H3K4 methylation levels are tightly regulated because of their important functions. So lysine-specific demethylase 1 has been found to be a member of the MLL transcription complex. LSD1 catalyzes an oxidative demethylation reaction with H3K4 methyls and thus counteracts or balances the enzyme function of MLL. Both MLL and LSD1 are essential for hematopoiesis, showing balanced cellular H3K4 methylation, and this is crucial for normal hematopoiesis. Hematopoiesis is, for my understanding, maturation of blood cells, their cellular division and their maturation. For information only, H3K915, so this is another position within the histone H3, and other proteins such as DNMT116, which is a DNA methyltransferase, which generates epigenetic DNA markers, were reported as substrates of LSD1. Notice this is a figure for information only, and again it's because it's a little heavy, it's a bit complex. So what we've got is the transcription complex, and what happens is initiation of a gene reading by changing the methylation, by changing the signal on the histone, and notice this is a transcription event. This is a classical depiction of a transcription, which is the gene unwinds, and here we go, it's being read and turned on. So this is the brown line is the gene, and it's currently wound around the histone octamers. Various things have to get together and modify the signals on H3 and H4, driven by a transcription factor, and notice P53 is in there, if you've ever come across P53. P53 is an oncoprotein, so P53 can be mutated in certain cancers as well. In fact, quite a lot of cancers are generated by P53 mutation, which would certainly affect a P53 mutation might affect driving of this gene and then dysfunction. All MLL translocations identified to date, that's all mucking ups of the MLL gene, identified to date, produce MLL fusion oncoproteins. In other words, proteins get fused together within this gene sequence when they're translated, and then instead of being separate proteins, they get fused together, and this can disrupt the methylation status of the histone tails at K4 and K9, and therefore that's going to change the signal at those genes. Outcome of the disruption appears to be linked also to local local DNA methylation status at potentially affected genes. In other words, there appears to be what's called an epigenetic crosstalk. In other words, the epigenetic signals by methylation at the MLL, at the gene that MLL works at, and the DNA methylation at that gene are both affected, or appear to be both affected. Often these fusion partners can also recruit what's called DOT1L, which is a histone H3 lysine 79 methyltransferase, and this causes these fusion partners wouldn't normally recruit this methyltransferase, but when they are ill-made by effectively translocation or mutation of the gene, this causes aberrant H3K79 methylation at the MLL gene, target gene. And overexpression of these genes eventually leads to initiation of a leukemia. Excuse me. Pharmacological inhibition of DOT1L has been found to block proliferation of MLL rearranged leukemia cells, and that shows effectiveness of targeted epigenetic therapy as well. So DOT1L in its own right, rather than LSD1, is also found to be a epigenetic target. So the note here says, think, mentioning the MLL complex but without extreme detail. How could you précis or sum up the last 3 slides with some key points in a few sentences? Okay, so I want you to take that information and condense it down to a few sentences. That's all that will be required, but the discussion gives you a sense of what can occur within leukemia. If you need to do any additional reading to do that, by all means do so, but the slides should provide you with enough fuel. You can go to the original JMedChem journal here to see a different version, not a different version, but the full version of this. Okay, let's cut to the chase. So we're going to look at LSD1 as an anti-cancer target. So what's the background to inhibition of this enzyme? Relatively recently, it's been found that monoamine oxidases, MAO-A and MAO-B, are FAD cofactored oxidases. And the inhibitors of these enzymes, which you probably have met before in CNS physiology, stroke pharmacology, inhibitors of these enzymes are able to inhibit the very closely related monoamine oxidase like LSD1. So what that means is MAO-A and MAO-B, which are FAD cofactored, are very similar to LSD1. Trenylcypramine is what they call an archetypal compound. It's an early inhibitor known to work at the MAO enzymes. And notice that there's a bit of stereochemistry around that three-membered ring, and also notice there's a three-membered ring. What the three-membered ring is actually doing is constricting the conformation of the NH2 relative to that phenyl group. Phenelazine, or nardelazine, is a different class of inhibitor. It's a hydrazine, which is not a normal functional group found in drugs. And then selegiline, or deprenyl, which you may have heard of, is also a monoamine oxidase inhibitor. And so something is happening with this functional group combination, the three-member ring and the amino, the hydrazine around here, and then the nitrogen and the triple bond around the terminal part of the deprenyl. So these inhibitors inhibit the LSD1 as well as monoamine oxidases A and B, and they have some selectivity between A and B. But so as a result, if we're going to look at these type of compounds to inhibit LSD1, and they are known inhibitors of LSD1, we would have to avoid the off-target enzymes to reduce potential side effects. So when we're developing compounds against LSD1, because MAO-A and MAO-B are closely related and work in the CNS, we would want to try and avoid inhibiting these enzymes. So we're going to go through the design journey. We're going to take a paper by Wu as the example of the case study. So they took their early lead from tranylcypromine, or TCP, as many people have. A lot of people have used this starting drug as a scaffold for drug development. So they took tranylcypromine, and notice that it's micromolar at all of the enzymes, with some selectivity towards MAO-A and MAO-B, but a reasonable nod towards LSD1. What you notice is if you increase the NH2 to include a heterocyclic ring, a CH2 and a heterocyclic ring known as a piperidine, and they played to get to this structure. It wasn't straight to this structure, but if you go to this structure, you notice that there is a 250 times increase in activity towards LSD1 and 1500 times more selectivity for LSD1, which is a good thing. So that means we can differentiate between LSD1 inhibition and MAO-A inhibition. Knowledge of other compounds from other groups working in the area led them to believe that they could extend the amine of TCP as they did, and computer-aided molecular modelling supported the changes going from 1 to 2. In other words, they used the crystal structures of LSD1. They probably also used the crystal structures of MAO-A and MAO-B, although this isn't mentioned in the paper, but I would suspect that would be the case because it's something I would do, and these are freely available, so the X-ray structures of all these enzymes are freely available for drug developers. Now if we look here, we've got this middle bit, and we're saying that we can extend this end and we can extend this end, and these are known inhibitors of LSD1. And we've got this structure extending at this part of the structure from the hydrazine-like part of the hydrazine moiety, and here is a heterocyclic ring at this end and one at this end, and that's what this is doing. It's being like these. So if we now look, we're going to change from 2 to 3. And why have we done that? We've done that by replacing the cyclopropyl ring with a pyridine ring or pyridyl ring and an O. That allows us to attach, we'll talk about this in a minute, and attach this various groups to this. It also gives us an aromatic ring that we can do maybe other things with. And why would we do this? Well, it removes the problem of chirality. So if we look back here, we've got very fixed chirality at these 2 points. These are specific chiral centers, and with this drug as well. And we're replacing that to an achiral drug, and that narrows the problems with any legislation or fine drug legislation later on. It increases, so it removes the chirality and increases the ease of synthesis of analogues for optimization of activity as well. Docking studies of a model of LSD1 suggested that addition of an extra phenyl group choice of the pyrazine was for rapid synthesis of 4. So here we are. So the story goes that docking this into LSD1 showed that we had some room here, but the difficulty of putting a phenyl on the pyridine, initially they wanted to move the drug development along, so they made a pyrazine instead, which is this part is much easier to synthesize, and you've got a comparable compound. It's comparable activity. So that was for rapid synthesis. But what they found was when they changed or found a suitable synthesis for pyridines, including an aromatic ring here, that gives them a chance to put functional groups off of here as well and to get rid of the pyrazine. And they notice also in this particular compound that we have increased by at least tenfold the activity towards LSD1. However, from general structure-activity relationships, and remember back at the previous lecture I said about QSAR, just an understanding of what structure-activity is about, just a quick look at those slides will help you follow what's going on here. So general structure-activity relationships and synthetic considerations, it was decided to pursue 5 as and its analogues as an extension of the drug development program. So the rational design went like this. This is the LSD1 complex with the pyridine docked in to the active site. What they noticed is that there's space in the enzyme at this position. It's hard to, it was harder initially to introduce an extra phenyl, so they made a pyrazine, which just happens that synthesis is easier. But there is a problem with pyrazine synthesis, and that is that it's very hard to make asymmetrical pyrazines. In other words, 2 different groups on here. It's very easy to make symmetrical ones where this is the same as this, but it's actually difficult to play with one side versus the other, which is why they ended up going back to the pyridine. So the lowest energy mode of docking of compound 3 with LSD1 active site model. Note that there is a key hydrogen bonding interaction between the pyridine ring in both cases and D555, which is a carboxylic acid containing side chain. The space available suggested elaboration for rational design and introducing an extra phenyl group. And that's what they did. But to do that, they had to come up with a sensible synthetic scheme. Now, you are well aware as a cohort that learning metabolic cycles is a requirement for things like physiology and pharmacology, maybe if you're to discuss those things in those scenarios. In this case, this is a point where I would rehearse and practice this slide because the chemistry is needs to be understood. So what they started with is a dihalo substituted hydroxypyridine. This is going to give us a chance to do this here at this point, and we'll talk about how that happens in a moment. And this difference between these two halogens is going to allow us to put selectively one group on one and then a second group on the other because of the reactivity difference between these two positions. So what's this reaction all about? The first step is an etherification. So in other words, you make an ether linkage between a protected piperidine alcohol. It has to be protected because coupling strategies would, if that wasn't protected, an NH and an OH would react at this OH equally, or at least both possible. So you protect the amino group by hiding its NH away temporarily. Then you make the attachment to make an ether, and we met ethers in the first year very briefly. You can, now that's all together, you can now react at R5, and the coupling here, this step, etherification, is known as a Mitsunobu reaction. The changing of halogens to aromatics is known as a Suzuki coupling. And then finally, once you've decided what you want on here, you can you can deprotect this group and get rid of the protecting group. Now, we're not going into the protecting group chemistry, even though in this paper it's specifically covered. But the fact that a protecting group is required is noted, and then that you can remove that when you want to release the compounds you want to test. What that did is it allowed this group to play around with these aromatic compounds as to what they had around the ring in various positions on both separately, because one is R5 and one is R6. And you could connect the aromatic groups up separately to the pyridine, and then you had different groups on these positions. And by changing these around, but you can see that this has stayed the same, although there was a little playing around with this. We can fit into the cavity, we can get our specific recognition with D555, and we can extend particularly into small regions to fill the pocket within the enzyme in a selective way and to generate a potent compound. This compound 17 was found to increase the cellular levels of H3K4-methyl-2, which means that it's a lysine demethylase inhibitor and stops the methyl group coming off from Me2 to Me1, and it's selective against leukemic cells with an MLL translocation, and there's no effect on healthy cells, therefore it's not toxic, even though LSD1 is required in other cell lines, which is good. Yeah, if you think about the body is using LSD1 in other tissues, not just in blood cells or blood hematopoiesis, and it's selective against LSD1 versus MAL. So as a drug journey, it's covered all of the aspects that we talked about throughout the course, which is, let's go through them quickly. It means going all the way back to the beginning, I'm afraid. Right the way back. So, we identify a target. We can get that target from genetic information or from understanding the role of particular proteins in a therapeutic situation. Sorry, not a therapeutic situation, a disease state. We know really what epigenetics is about, and we think that epigenetic modification can come about with therapeutic intervention, and it's not going to be so hazardous to the body, even though it's critical in the body's normal function. We understand at a molecular level what's going on. We know that the histones are complexly modified by post-translational modification. So the more that we understand this, the better we are at making selective drugs. We know the positions on the histone tails, for instance. We know that the signal can be written, read and erased, and an understanding of all of these processes will help us develop safer drugs. There is actually a precedent because for histone demethylation, and that is that histone acetylation and deacetylation are already proven epigenetic targets. So histone deacetylases are now in the clinic for treating certain cancers, and certain HAT inhibitors, histone acetyltransferases, are also useful in treating certain cancers. But HDAC inhibitors have got the lead. Let's understand the methylation. So what do we do now? We know that there are enzymes involved. We even know that one of the enzymes is a slightly different class to the others. So we understand the pathway. We understand it at a molecular level, such that we even know where within the active site of the enzyme the action is going to be, and that's close to the FAD. There is more knowledge about the FAD and its mechanism of removing the hydrogen for this step here. So there's quite a lot of chemistry known about this first step. That helps us understand this chemical interaction here. We're using X-ray crystallography to move the drug development program on. And here is an example of a mimicked system which helps us think about this active site and helps us think about how these are arranged with respect to the cofactor and how our drugs might be as well. And in fact there are peptide analogues of H3K4 which are known inhibitors. We now start to understand a little more about the fact that the LSD1 works in more complex situations, and that may be of value later on when we're really talking about understanding exactly what's going on. And we've only just started to scratch the surface of how LSD1 actually works. And this is an example because this is a relatively recent publication in how elaborate LSD1 actually is. And this is why people really want to understand how it works in the cells. We understand how signals can be read. And by the way, chromodomain and bromodomain blockers are also anti-cancer targets. So stopping the recognition of a methylation signal or an acetylation signal, bromodomain inhibitors and chromodomain inhibitors are also being exploited as anti-cancer targets. We understand leukemia as a particularly important cancer, particularly in infants, where it can be pretty destructive. 75% of acute leukemia in infants occurs because of histone methylation dysfunction. And that's because of the gene product from MLL, which is a multi-protein complex. So the MLL gene reads for several proteins in this complex, and when that goes wrong, it generates gene products that go off and interact with their gene targets, because the MLL protein complex is something that works at genes. So it's a gene product that works at genes. And this subtype of leukemia shows very poor prognosis. So we need something to be able to treat this. What is the MLL protein, or multi-domain protein? It's a 4,000 amino acid protein, and that's a big protein, and part of that protein has a set domain within it, and that is catalytically active as a histone methyltransferase. So understanding what's going on and its balance within the normal process, MLL and LSD1 working together are required in a normal operation of methylation and demethylation at normal blood cell function. Hematopoiesis is crucial. When that goes wrong and you get a misbalance, particularly from MLL translocations, when this protein gets buggered up, as it were, excuse my French, it's important. This is just to show that the MLL complex, this big protein interacts with many other components, menin, MOF, WDR5, and it works at an initiation and an elongation of its target genes. And when MLL goes wrong, it drives target genes in blood cell hematopoiesis to proliferate blood cells, white blood cells, which is what leukemia is. So now we understand, we also can understand by looking at specific diseases, and once we understand methylation, histone methylation is important, we also come across the fact that other enzymes can be important in the process and become drug targets in their own right. A simple explanation of these slides is worth it, and we're talking about maybe 5 or 6 sentences, no more than that. A few sentences. Then we go into understanding the inhibition. So what we know is that monoamine oxidases are very closely related to LSD1. And actually, a lot of the initial work was done on plant enzymes that are monoamine oxidases before they started to look at MAO-A and MAO-B in humans. MAO-A and MAO-B are drug targets in their own right for treating central nervous system complaints, and LSD1 is related to these. These 3 drugs are known to inhibit MAO-A and MAO-B selectively, so what that sets is a precedent for knowing that we can design drugs that are selective between MAO-A and MAO-B. If that's the case, we can probably design drugs that are selective against LSD1 versus MAO-A and MAO-B. So we get selective where we want it and no action at MAO-A and MAO-B when that's not required. And that helps reduce side effects. So this is typical. The reason why I chose Wu's paper is that this is a typical process of going from TCP, or tranylcypramine, which has this stereochemistry around a three-membered ring, and its mechanism of action is very well understood at MAL A and MAL B, and now LSD1. And an understanding from other compounds from other people, in other words, chemical intelligence. What are other people doing? How are they dealing with it? How are they making inhibitors? And notice one of them is a TCP analog. This one's a phenylhydrazine analog. So you can use the information from both courses to inform your drug development program. We can play around, and we choose the chemistry to play around in where it's easy to make a rapid progress with a drug development program. So we can make, if you look at this paper, which I suggest you do, by the way, certainly download it as a PDF and see how the group used pyrazines to move this side of things together. So what they did is they made pyrazines that were symmetrical. Both phenyl groups were changed at the same time, to have a quick look at those. But really what they wanted is to be able to change one and not the other. So they had to go back to the pyridine chemistry to do that. And they did that by, they knew that they needed to do that by looking at the X-ray structures of the LSD1 enzyme, and then modeled by docking the compounds of interest and looking at the spaces that were available by using computer-aided molecular design, the spaces that were available within the active site. Now, don't think that you need to reproduce diagrams like this. You can do your own cartoon schematic diagram of this that's much simplified. It is worth, however, noting that there's that specific hydrogen bonding interaction. A synthetic route that has, so this is where a medicinal chemist, a synthetic chemist comes in. They can design a synthetic route that has broad applicability to be able to change R5 against R6 with some ease, and that's why they've chosen this as a starting material. This was a very key step forward because it allowed them to bolt together the bits that they wanted to be able to get a range of R5s and R6s. In fact, this structure, just with an R5 and an R6, is a very important structure to memorize. And then eventually you can see that they settled on one compound, compound 17, which has got a nitrile group on here, which we met in the first year when we were talking about aromatic chemistry, nitrogen containing compounds, and a methyl group at this position, and that fits into this site nicely, and the cyano group fits into a pocket down here, or comes into a linear channel here, and this is a very snug fit. So we've got a selective inhibitor, it's not toxic against other cell lines, and it's selective for LSD1 over MAL. And that's the journey. I know it's a tough topic for you, but as I mentioned before in the lecture, it is now master's level that we're pitching things at. Try and find a way of telling a good story for the case study that shows that you understand all of the concepts that we've used before. The paper talks about screening, so we talked about screening being important in drug development, selectivity, not toxic, off-target, avoiding off-target targets, understanding the pharmacological, physiological background of the disease that you're trying to attack, like leukemia, and then using all the tools required along with synthetic chemistry to get to your final target compounds to get your candidate molecule, and that's almost where they are now, at a candidate level. and thanks very much.